I've called this, I've been studying Margaritifera now for 30 odd years. And I suppose so much has happened with this science in that time that it's a bit like being asked to talk about nuclear physics or something. Where do you start? How much do you include within a 40-minute talk? So I'm going to do a bit of an overview of the research, the science of Margaritifera study in the last 30 years, and perhaps try and look how it's evolved since before 1990 and through the 2000s and into the future, and just pull out a few examples of what's been going on throughout Europe and work carried out by the Margaritifera community as a whole. So Margaritifera in various guises has been around for a very long time. And virtually unchanged. The top left photo is of a fossil from the Isle of Wight. We call it Unio valdensis in inverted commas, but it's so similar in shell morphology and structure to the modern Margaritiferates, which are shown there, the other two European species, Margaritifera auricularia in the center, and which formerly lived in Britain, certainly till the Middle Ages. And the one below is Margaritifera margaritifera. So they have a very long history going back 150 million years, little changed. We know they have a complicated life history, which involves young salmonids as an intermediate host for glochidial development and for juveniles to drop off. We know that they are highly demanding species, particularly regarding water quality and water geomorphology. And we know that they're a very long-lived species, certainly in Britain and Ireland for just over 100 years, but in the Arctic to a lot longer. Its longevity has compounded really the difficulties of some of its conservation, particularly as its populations can dwindle over a long period of time. So, you know, extinction is a slow process in a lot of populations. This would be our ideal scenario, I guess, of what a Margaritifera population should look like and the sort of habitat it should live in. This is probably our best river in Ireland, in the west of Ireland. Lovely open landscape, no trees, very little, in fact, no agriculture apart from some rough sheep grazing. Lovely flow on the river, good nice mix of substrate, great big beds of mussels, or seemingly doing quite well. And the mussels are present all across the river, from one bank to the other. So that sort of habitat, let's go back one. That habitat might be considered quite typical of the western and northwestern parts of its range. That's what you might expect to see in Western Ireland, quite a lot of Scotland, certainly Norway, and into the Arctic. However, through much of its European range, it's living in much smaller streams and rivers, very often running through forest. The mussels tend to be, the populations tend to be quite small, whereas the ones in, might be several million in the example I've shown from Ireland. In these sort of situations, you might only have a few tens of thousands, and they might be restricted to the areas where there's preferential flow. But in both scenarios, this is the sort of what you would want to see in a functioning population, in that you would have lovely clean sands and gravels full of juvenile mussels showing a complete size range from, looking at the bottom one, that's probably running from 100 years down to about 2 years old. So that would be a nigh on perfect population demographic profile. But as I'm sure most of you know, this is now a great, a scenario like this is a great rarity. And there's probably only a handful of rivers throughout its world range that would have such beautiful conditions in population demography. So I'm going to, whilst I'm going to try and keep some sort of flow to the talk in terms of how the science has evolved a bit over the last 30 years, it's going to be a bit of hopping around, so please bear with me. Let's have a look at the maps from the two Conchological Society atlases, which sort of certainly at the time reflected the knowledge of the species distribution in Britain and Ireland. You can see from the 1976 atlas on the left that there were very few post-1950 records at that time, just a handful dotted from Devon up to northern Scotland and half a dozen records dotted around Ireland. By the time of the publication of the 1999 atlas, the range is unchanged. I think we all know that the range is confined to the non-calcareous geology in Britain and Ireland. However, there's been a lot more records filled in, and a lot of the old records were updated to living specimens. And that range stands today, apart from maybe sort of the central and western part of Ireland. Let's see if I can just put my cursor on. Around here, most of those records now are updated to living populations and indeed support some of the best Margaritifera populations in Europe. So, and to some extent, particularly with the Kearney 76 atlas, most of those records were based on old ones gleaned from publications in the journal, but to a greater extent, they were records based on data freely given at the time by Peril fishers, which is not mentioned. So then we sort of move on to the 1990s. There became a sort of, with the advent of the European Habitats Directive in 1992, there was a sort of impetus by the 3 country conservation agencies in Britain and NIA in Northern Ireland and Parks and Wildlife in, or the equivalents at the time, to establish which rivers should be chosen for Natura 2000 sites, i.e. special areas of conservation, SACs, because the information required to make such designations was generally very poor. So that it certainly initiated a lot of survey, certainly in England, Wales, Scotland and Ireland. But at that time, really, we were just looking for really quite simple surveys to establish the size of the population, where they were, and where possible to investigate if it was a recruiting population or not. And as a result of that work, SACs were designated for each country. In Ireland, there's 27. I think there's 27 in Scotland as well. There's 1 in Wales and 3 in England and 3 in Northern Ireland as well. I'm going to take a step back. It wasn't just some of us going out and doing surveys. And I'm going to take this opportunity to have a nod to two pioneers of Margaritifera science, certainly for Britain, and that's Professor Mark Young, now retired, of University of Aberdeen, and we call him Dr. Bischoff, no one even knew his Christian names, Wolf Dietrich Bischoff, from Germany, who certainly by the early and mid-80s had recognized that Margaritifera was in a lot of trouble throughout its European range, and that much more conservation and research effort should be put towards this species. So I think we owe a great debt for their pioneering and pioneering work and foresightedness. With the increased interest throughout Europe and requirements from the European Habitats Directive, it sort of culminated at the end of the 1990s with this meeting in Hof in Germany, where there were participants, 235 participants from at least 15 different countries, that might be more actually. And everybody got together and drew attention to the wider scientific and legislative community, that much more effort was required to conserve this species, and it was a species that was critically endangered and not just in general sort of decline. So that acted as another springboard for a lot more work on this species. And since that 2000 meeting in Germany, I think it's right to say that until COVID, there was at least one international conference in Europe every year on some aspect of science for the freshwater pearl mussel. I'll mention the EU LIFE programme, which is what really got a lot of the original projects moving. I'm sure most of you are familiar with EU LIFE. And there's been, to date, I think there's been 42 projects covered by EU LIFE on freshwater mussels, the majority of which have been on Margaritifera, with 2 or 3 extra ones on Unio crassus. And they having, and that's, I think as a result of those and other work, the freshwater pearl mussel has become the most studied mollusk species in Europe, and is certainly the recipient of by far the greatest investment in funding. It outweighs all other mollusks by a very large factor. And despite all that effort, both high quality science and funding, the species is still far from secure. I think we would still consider it to be critically endangered and in decline. In the UK and Ireland, there was early LIFE funding. Start kicking off with the Life in UK Rivers series from 2000, which various of us contributed to, and really provided a synthesis, a much needed synthesis at the time, of the ecology of the species and ideas at the time for how the species should be monitored. A lot of this was led by Mark Young and his team of students and ex-students, which includes Peter Cosgrove, who's with us today, and Lee Hasty, who I'm sure some of you are familiar with. So that provided those manuals provided a very sound basis from moving forward, for monitoring and conservation of the species. In the mid 20-teens, we had the Pearls in Peril project, life project, which was a pan British project. There were one river in Wales, one in the Lake District, and a few rivers in Scotland were included in this project, mostly looking at catchment management and habitat restoration for the species. And lately, in the sort of latter half of the 2010s, we had the Kerry Life project in Southwest Ireland, which was focusing on two of the best mussel rivers we have. Threats to the pearl mussel, really, you could have a list as long as your arm. This is just some of the many in their various wide and narrow guises, but really sort of drainage, catchment intensification and habitat loss is the prime threat to the mussel. But all of those others are very real in various parts of its range, some to greater extent than others. I know that Scotland still regard pearl fishing as a serious issue, whereas throughout a significant part of its European range, other issues are more important, such as forestry and even invasive species you might be surprised to know, such as introduction of fish that don't belong in the river and are not hosts for the species, and so on. Let's just have a look at some of these. As I said, I drew attention to catchment intensification, habitat loss through drainage. Drainage of upper catchments, the sort of wetland areas of upper catchments, is a particular problem. Because those upper catchments are no longer acting like a sponge to keep the water back and allow it to come out at a constant rate. There's agricultural drainage in the middle at the top. Then we have conifer forestry and peat digging alongside rivers. Peat digging in the bottom left. This is against one of our best SAC rivers in Ireland. Peat digging goes right up to the edge. Conifer forestry right up against Margaritifera rivers, and then the clear felling with all its associated problems. And the bottom right is actually it's industry, if you want to call it. It's a mink farm butting up against one of the top 8 mussel rivers in Ireland. Not so much a problem to the adjacent river, but a lot of the mink waste is spread around the catchment, which is far from ideal, obviously. One of the main features of drainage, of course, is the release of solids into the river, and I think in the early 2000s, siltation of Margaritifera rivers and clogging of the substrate was regarded as one of the main problems with the decline, accelerating the decline of Margaritifera, i.e. the infiltration of the sediment was causing anoxic conditions and the subsequent loss of all the juveniles and thus juvenile habitat as well. It's all pretty horrible. We've seen pictures like this many a time. As with these, this is nutrient, mostly phosphorus, causes the growth of extensive filamentous algae. If it's an ongoing problem in a Margaritifera river, it's normally because the phosphorus levels are constantly too high. Although you do get accelerated blooms with runoff as well. Macrophyte growth is also a bit of a problem, which is a bit of a conservation dilemma, because Ranunculus penicillata is actually a protected species, yet it does cause problems where it grows in Margaritifera rivers, because it attracts sediment and then it grows and you get more sediment and you build up these muddy, sandy banks. So again, loss of habitat. But really the main problem that is recognised now is flow. Again, this is one of our top 8 rivers in Ireland for the species. And this, you know, it has become subject to extreme low flow events, and the mussels migrate to very small parts of the channel where there is suitable habitat to try and avoid these drought conditions. I'm going to come back to drought and climate change later on. As I said earlier, so constant nutrient input is a result of, you know, continued elevated levels of phosphorus. But when you have low flow, very low flows for protracted times, even in the most pristine systems, which have scarcely any nutrient and scarcely any sediment input, you can still have severe algal decaying matter from algae, having quite a dramatic effect on both mussels and the habitat. This goes back to an event in 2012 in one of the best mussel rivers, and you can see that both juveniles and adult mussels are climbing out of the sediment to try and find their way into better flow. And then continuation, they dig themselves out completely, and then it's quite a die-off of mussels of all stages of the life cycle. I think a very good example of how flow is a key issue. This is a series of photos from Arctic Finland. So it's up in the Arctic Circle. You have a very large lake which is frozen for a very significant portion of the year. This is late May. These photos were taken. It has a fabulous mussel population, again, wide demography, just what you want. But even in these wonderful conditions, the mussels are only occurring in probably the 1.5 kilometre outfall from the lake, where the flow is right, where the substrate is stable at all times of year. And because of the very large lake, which is frozen for a lot of the year, the flow is nice and constant as well. It's not subject to great fluctuations. And you see in the bottom right, this is how our Finnish colleagues survey for 2-degree water. They just take their trousers off and paddle around. But of course, you know, all these problems are in combination, and this is the classic vicious circle of nutrients, inadequate dilution, sediment runoff, rainfall, and it all goes around in circles. So all three of these issues are acting in combination to not help the conservation of the species. One of the look a little bit at monitoring and how we sort of moved on in the early 2000s, really sort of following to some extent the proposals from the Life and UK Rivers manuals. I think most countries set up permanent transects as a means of assessing population size, particularly in large populations, and to use them for monitoring. In England and certainly in Ireland, we chose to use transects, quadrats in transects which flow across the river from one bank to the other. The very large rivers, particularly some of the ones in Scotland, don't really lend themselves to transects all the way across. So a lot of them tend to use sort of longitudinal parallel to the bank. However, they've proved to be useful and not useful. This is an example of one transect on a very good mussel river in Western Ireland. And I just draw your notice to Transect 4, which I've highlighted, because I'm going to come on to that in a few slides' time as well. But you can see, I think we have big numbers of mussels going across the transect, but it's varied considerably from year to year. Now, this is with the same pair of surveyors, Evelyn and myself, using the transect as well laid out. There's no doubt that it goes across exactly the same place. Yet we've had very significant variations year on year, and there's clearly a trend of increase, which you might ask, well, why is this? And I'm not really going to go into the reasons, but it's it makes it, one question is how useful transects are for assessing big populations, and is that enough for monitoring the species? And the answer is no, we need a lot more information. Running in parallel in harness with the permanent transects, where one tries to do a lot of demographic work as well. It's an invasive technique, and I don't think any Margaritifera scientist really likes doing it. However, it is essential to have these data as to keep a handle on the health of the population. This is two rivers from Western Ireland. We'll look at the bottom one. That's a classic example of the way a Margaritifera population declines. So we have the y-axis is percentage of mussels in those size classes. The x-axis is the size class in millimeters. Sorry, I should have put that on. But you can see in the bottom slide, there's been a complete shunt in the population size distribution from 2004 to 2019, such that there is no, it would give you the impression there's no juvenile recruitment at all, or certainly no juvenile survival, which is true. In the top one, we have a bit of a wider, we have 3 years worth of data. And you can see in 2001, the population was doing very well. There were a very large proportion of juveniles. This is the blue histogram. However, by 2004, that had flipped, and the population had gone much more to the right with very few juveniles. However, by 2019, there's a considerable recovery in the percentage of the juvenile population. And what I think this really highlights the need for doing this work is that you only need one major pollution event or some other catastrophe to completely wipe out 5 years worth of juvenile recruitment. And it takes a long time to recover from that, even in a river with the quality that's capable of supporting a fully functional population. So sort of moving on through time, I think we recognize that there was a need to do much more investigative work to identify specific problems. And two tools which we particularly favor is the use of redox potential and for looking at the river velocities. So sort of collecting more physical data rather than focusing just on the mussels themselves. Redox is really, it's a surrogate means of looking at the oxygen levels within the riverbed substrate itself. So you're using 2 electrodes, one in the water, one in the sediment, and you are looking for the difference between the open water and the substrate. The less difference, the greater the oxygen levels in the substrate. And the graph on the right shows the relationship between redox level at a depth of 5 centimeters in the substrate, with the smallest mussel taken from quadrats within those rivers. And there's a very strong pattern there that juveniles are surviving, living only in substrates with the lowest loss of redox, i.e. the highest levels of oxygen in the gravels. But it's a very useful tool for monitoring. It's easy to do, you can get a lot of readings in a day. And this is looking at flow velocities of the water, both at near bed, excuse me, and at 60% depth in the water column. This is going back to the Transect 4, which I showed you earlier, where we were looking at changes in just the numbers of mussels per quadrat over time. So this is just looking, the red histogram is the numbers of mussels in each 1 meter quadrat, and green and the blue are the velocities related to water depth and the percentage loss in redox. And there's quite strong correlations there. You can see with our quadrat number 6, the velocities are low, and the numbers of mussels are low, i.e. so that water velocity is not high enough for large numbers of mussels. Conversely, if you go to 17, 18, then you've got low numbers of mussels, but very high velocities, too high, so you're getting riverbed scour instability. But again, it's these combinations of looking at all the different stressors, and we can combine those data to try and get a much better picture of what's going on in the river and what might be causing the problems. Moving on, you might remember a couple of years ago we put a paper in the journal of Conchology on this new technique for habitat mapping, for, shall we say, total population assessment, where we were looking at transects across a very good river in England, and looking at transects every 10 meters and doing a wide range of parameters, including the numbers of mussels, obviously, per quadrat, but looking at all these other environmental parameters as well. So we're collecting a huge amount of data, at the end of which we were able to assign each quadrat per transect to whether its habitat suitability was good, or whether it was potential, or whether there was none, and similarly with the condition the habitat was in. Using GIS, we were able to sort of start to compile layers. This is looking purely at habitat, the green being good, the red being poor or no Margaritifera habitat. And remember, this is short, this is small scale. These transects are only 10 meters apart. And then you can start to work the magic with the GIS and start to interpret all the intermediate habitat to get a very clear pattern of the distribution of Margaritifera habitat in the river. So what does this tell us? Well, okay, it does identify the total amount of habitat, which even in a good river might be as little as 30 or 40% that is suitable. It certainly identifies the condition where it might be too scoured, where it might be too, where you might get sedimentation, and so on and so on. And it provides realistic objectives for conservation. It certainly identifies the best and worst place for captive bred mussels, which I'm going to go on to talk about, so that you have a much better picture of where you should and shouldn't be putting them. And because we're collecting all these data systematically, we can do a lot of cross interpretation for the sort of multiple stressors. And we can use them to relate to the juvenile population assessments as well, where we were assigning every juvenile quadrat to one of the categories of whether it was good habitat, good condition, and so on, the sort of 9 combinations. And just looking at these two, this is the top one is for good habitat, and the system works. The juveniles are in the good habitat that's in good condition, to a lesser extent in the good habitat in moderate condition, and not really in the good habitat in poor condition. And if you look at the unsuitable habitat graph at the bottom, you can see that there's hardly any mussels at all, let alone juveniles. And this technique, we're pleased to say, is now being used in several places in Europe to assess populations, particularly for, where they're going to put captive rear juveniles. And I think it's just been proposed as a method for some work that's going to be done in a conservation augmentation scheme in a river in Wales. So we're pleased that that's moving along. Genetics, we've started, I think, throughout Europe and North America now. Genetic studies have become a very important part of freshwater pearl mussel conservation. That's an extremely useful tool, particularly for not only for wild, but particularly for captive bred mussels as well. You can see that there are strong genetic differentiation within populations across the range, particularly between differences between Southwest Europe and Northern Europe, North America. We did carry, a couple of years ago, we carried out and published on some work that had been done on some of our Irish populations. We looked at 9 rivers, which included the River Nore, which just about supports this population known as Margaritifera duravensis. And we tried to get a wide geographical spread to kick this off. This is working with our colleagues in Munich. And the Irish populations that we've sampled so far have fallen into 2 main conservation units, a salmon-dependent western cluster and a trout-dependent central and eastern cluster. Although Margaritifera duravensis was found to have the highest genetic divergence, it really fell within the group of the other central and eastern pearl mussel populations, and really can be considered to be part of the Margaritifera taxon. There was high genetic diversity with Western populations, suggesting that it's quite a hot spot in terms of Margaritifera genetics, whereas the Southern and Eastern populations, much lower variability, but greatest differentiation and uniqueness. So it's thrown up quite a lot of information as to how we should move conservation of Margaritifera in Ireland forward. And we will be continuing with sampling further rivers, hopefully next year. A lot of legislation's happened over the last 10 years, certainly. Of particular importance is this SEN European standard. About 25 of us got together over about 3 years at various meetings to try and produce this document. European standards are normally based on weights and measures and all of that sort of stuff. So this was the first standard that was developed for a living organism. So it's quite a groundbreaking thing. And, it has sections on virtually everything that affects Margaritifera and what should be looked at when you're trying to assess plans and projects on Margaritifera rivers. And it's certainly been significant and has resulted in a much more standardized approach to assessment, particularly when there's commercial and forestry and drainage activities going on within in a catchment. You can, I think all EU countries signed up to this, so it's a stick that, and the UK signed up when you were still there. So it's become an extremely useful document for conservation. Captive breeding has become quite a major issue, an important issue for Margaritifera conservation, certainly over the last 10 years, although in some places, particularly Ballinderry in Northern Ireland, it's been running for perhaps 23, 25 years. But when setting such a facility up, there's multiple considerations. And really, I think all the work that's gone on so far is demonstrated that a very large financial investment has been required, both in terms of infrastructure and staff. And to some extent, this is why a lot of the EU LIFE projects have funded breeding stations. And most countries now have either attempted or they do have some sort of Margaritifera breeding station set up. There's a lot of issues to consider, some of which I've put there. The one perhaps most of you will know about is that the Freshwater Biological Association on Lake Windermere, which has been running since 2007. It was set up basically to act as an ark for threatened and declining populations, but also to try and produce, to captive breed from these populations. They've had their successes and they've had their failures, like every breeding station has, but they've certainly done very well with 2 of the Cumbrian rivers. This is an example. This one actually is in Luxembourg, where they've had great success with being able to produce Margaritifera. But unfortunately, they're still trying to restore the river to a sufficient quality that they can start to really get these mussels back in meaningful numbers. And because they are so expensive to produce, they're precious creatures, and you do have to think very carefully about where you're going to put them back. And to this end, the Freshwater Biological Association have started this augmentation project of the River Ert in Cumbria, which is certainly one of the most famous mussel rivers in Britain. Read all the history books, but it'll tell you Queen Elizabeth got her pearls from here, etc. So there's no doubt that it was hammered by pearl fishing, but it had a huge amount of catchment issues such as forestry, sewage, abstraction, land use, and the list goes on and on. And the population had probably dwindled to about 500 individuals with no recruitment. So this project is being managed by Louise Levictoire at the FBA, and with the intention of trying to get some of their precious captive bred Ert mussels back into the main river. They've done a lot of homework and a lot of physical measurements to try and determine where these precious mussels should go, particularly looking at historic distributions, the extent and condition of habitat and area using the habitat mapping techniques, and so on. And you get me out of the paper. And to date, I believe they did a pilot in 2017 where they put about, I think, 70 mussels back, of which they managed to easily retrieve something like half of them. But since in 2021, they've put another over 1,000 mussels back in 2 or 3 different places, and using transponder pit tags this time, so that the mussels are more easy to find into the future. And fingers crossed that this is going to be an example of the way forward with restoration and augmentation projects. But the message is you still need to have a catchment and a river that is suitable to take these mussels back. Catchment restoration is becoming, across the EU, is becoming a major thing. In Ireland, we have this project called the Peril project. It's rolled out on a wider scale than EU life. It has been much less specific and gives the managers of the project greater flexibility. And really it's looking at agri-environment and trying to encourage farmers to do all the right things for pearl mussels and rewarding them for doing so. So there's something in it for them. And this project is being run in Ireland's top 8 freshwater mussel catchments. A lot of the work is addressing the drainage issues. So there's a lot of effort going into blocking drains. There's a lot of effort going into fencing to prevent poaching, and as you know, but they're providing cattle nose pumps to keep the cattle out of the river. So these are locally led schemes, and they ensure that the landowners participate in the projects and have ownership over the mussel rivers, which is if you've got them on side, you're getting a long way down the road. Let's look into the future a bit and try to. And we look at climate change and the green agenda. I think it's true that one of the major mantras of the politicians at the moment is develop more energy from wind power and plant more trees. But we obviously need to do the latter in the right place, and we do need to be aware of so-called greenwashing. Yeah, but this is a wind farm up on a peat-covered hillside in northwest Ireland, where we had an enormous landslide just recently, and it's heading towards a mussel river there, you can see, which is surrounded by forestry, conifer, citrus spruce plantations. So it's a major problem with trying to get wind farms in the right place. And in Ireland, that's particularly difficult because the land where they would be better is required for dairy agriculture. So it's a bit of a conservation dilemma and into the future. These graphics, this is again using new techniques. This is using remote sensing. And this is time sequences for effectively wetness of the land and transpiration. And so we've got a time sequence here. Sequence on the left equates to the water table level, and the sequence on the right equates to evapotranspiration levels. Now, in this catchment, the values are good because it's a very good, relatively unimpacted catchment. But there are two particular areas here which are showing up. This is up here and down here, where evapotranspiration is high and soil storage is low, and they are the two areas that are forested within the catchment. So this has considerable impacts on if we are going to have tree planting in mussel catchments. You know, we're trying, you know, it's going in the face of the green agenda, but really we don't want tree planting in good mussel catchments because it will increase water loss rather than benefit it. And this lovely picture is that river catchment that you've just seen. So the wetter catchments do have the better flow velocities and the more food for juveniles because of the saturated land. And summer rain reaches the river. it doesn't soak away. Almost there. Sorry, I'm aware I'm overrunning my 45-minute slot. It always happens. Trying to look at climate change as well, with its impact on mussel rivers. The hydrograph at the top shows flow for a very good mussel river in 2009 and 2010, where you do get periods of low flow, but you also get a lot of periods of moderate and high flow. So, which is what you would sort of, what we used to expect. But I'm going to look at this river now, and the bottom hydrograph is from this year up to October. And this, I've chosen 2021, 2019 and 2020 was not dissimilar, where you're getting a very extended period of low summer flow, followed by almost a cataclysmic flood event. If you look at the scale of the flow, the top one never goes above 2,000, and this bottom one is shot up to 7,000 megalitres a day at the end of October. So this is a trend we're seeing in a lot of mussel rivers, which will become a problem of extreme events, both in summer and in winter. I said that the destabilizing effect of this, we don't really know fully, and we won't yet, but it's of great concern. So to look into the future, I suppose survey of mussels now, I think, has become a very small element in their conservation. We do need to look at a much wider range of physical parameters associated with the pearl mussels, as you've seen, the flows, the substrate composition, so on and so on. Whatever is in the catchment is impacting mussels in some way. So the greater the information and the more cross correlations we can have, the better. I think there's not much doubt really that, you know, conservation money is sparse, let's be honest. So I think regrettably we will have to give much greater effort to conserving the most important spot populations, rather than spending the little money we have sparsely on population. I know it's not a popular thing to say, but spending money on populations that really don't have much hope, because the catchment issues can't be addressed. And really, you know, what can members of this society do? It has become an extremely specialist science now. But I think one of the things I would urge members is that if you live in pearl mussel catchments, is to keep your eyes and ears open. Every time we go to a mussel catchment, we see some problem. You know, we see a JCB in 5 meters of the riverbank, or you see yet another forest being clear felled. All these things, to some extent, someone needs to be alerted to them. You think, well, if I can go there and see that, why isn't someone saying it? So I think you can be the eyes and ears, and if you see pollution incidents, then, you know, the Environment Agency is obliged to investigate it. So there's a lot members can do. But I think really, as a society as a whole, I can't emphasize more how you just need to keep championing the conservation of this species. You know, people perhaps in Ireland here are sick of the publicity it's getting. You know, they sort of groan, oh, pearl mussels are stopping us from doing X, Y, and Z, but that profile needs to be kept at an extremely high level. Thank you. And this is a beautiful stamp we produced here a few months ago. Well, we did, but the Irish Postal Service did.
Thank you very much, Ian. I think that was a fascinating talk. If we can if you can cancel out of that, then hopefully people can be rejoining or revisualizing themselves. It clearly gives us a overall view of what many would see as a rather gloomy prospect. But I'm sure there are questions. Can I stop? Do you think the prospect overall in the future is positive?
Yeah, but maybe for a very few populations in the range.
So you think that in the long term, it'll be sort of common but restricted, common locally, but a very restricted range of sites?
Well, the important thing is it's no good having this species unless you're having a recruiting, functioning population. So I think so much effort needs to be spent on rivers and populations where that is a feasible reality into the future. And if that means selecting just a few in, I don't know, Western Ireland, Scotland, Norway, then that is what we might have to do to guarantee the future of the species, augmented with captive breeding. Or else you're just going to have to captive breed the rest of the populations, and what will you do with them?
There's one question come up on the chat from Colin McLeod. Do you want to put it in person or should I read it out, Colin?
It's not really a question, it was just a comment really, that I think the inclusion of the name, or inclusion of the word Peril in the name is unhelpful, because it does encourage people to have a go at fishing them, and there is still a fair amount of illegal Peril fishing in Scotland. Some of whom are basically clueless people who've obviously heard about the name Peril, the Peril mussel, and I've even seen lake mussels being fished out, and people hopefully looking inside them and expecting to find pearls. So I think, I don't know if the society can think of a name that doesn't include Peril in the name, but maybe that's something we could do.
I blame the media. I blame the media for that. Wherever you get media involved in a Margaritifera project, they can't help themselves but bring up pearls. You can tell them you're blue in the face not to mention pearls, but they can't help it. They have to do it.
It may be an ideal, but I can't think it's likely that the name's going to change. Peter Cosgrove, you have your hand up.
Yeah, I was just going to hopefully slightly brighten the mood a little bit with the future element to it. Something that's happened in the last 6 months in Scotland is, and this is on the climate change and green agenda, is that some very large grouse moors, which are broadly speaking pretty bad for pearl mussels and river ecology because they're drained and burned, have been bought up by carbon sequestration companies with the aim of literally blocking all the drainage ditches and wetting things up. And so we have some enormous estates. And for those of you who don't really know this Scotland very well, most of the landscape is owned by a relatively small number of people. And it's been historically used for many years as a rich man's plaything for shooting. But that agenda is shifting, and the green agenda is coming up quite fast now. And I've seen 3 very large estates in pearl mussel catchments that are now going to be just basically given over to being wetted up and doing good things. So yes, the future for certainly several populations is quite grim, but there are some things changing, and certainly for us in our upland catchments, the peat dominated ones in particular, I think that we may be turning the tide, shall we say, which I'm quite comfortable with, if I'm honest.
Well, that's excellent news, Peter. I avoided talking about the situation in Scotland because I don't feel as I'm well enough informed with the up-to-date situation. So thank you very much for that.
Kerry, you have some comments?
Yeah, I was just saying on the freshwater pearl mussel, we did have a request as to whether we permit somebody to commercially farm freshwater pearl mussels so that he could sell the pearls, which is one of the more interesting requests I've ever had to try and formulate a response to. But also, has there been any mapping of the potential impacts of climate change on Margaritifera? I know that for some of the rivers in Wales I was looking at, drought was increasingly becoming a problem.
Yeah, and I think this is why we have to take all the modern technology that's coming to help us interpret current and future scenarios, and able to, and again, this is going to inform us as to conservation prioritization into the future, and re-wetting the land as amelioration.
Thank you. Does anybody else have any questions? Evan, you have your hand up.
Okay, can you hear me?
Yes.
Good. I'm new to the species completely, and I only know some rivers in central Wales. I noticed there's a big gap in central West Wales behind Aberystwyth in Ceredigion. I wonder if the mussel was ever found in that area, or is it?
Yeah, the yeah, sorry, I nearly named the river then. Yes, certainly there's quite a well-known pearl mussel river in West Wales, but they're all.
I didn't see it on the distribution map. There was a whole.
No, that's probably because they're not sufficiently up to date. But a lot of those Welsh rivers which supported or formerly supported pearl mussels, the populations were probably quite small, and they were only, and regrettably for catchment management issues, the populations only occurred on the very lower end of the river. So all of the catchment and all of its inherent issues is above where the mussels were living.
Yeah, so the watershed in that area is quite close to the coast. The river's quite short anyway. But thank you, thanks.
Has anybody else got any questions for Ian? I don't see any other hands up.
Ron's got his hand up, Tom.
Yeah, well, come on that one, yes. Can you unmute, please, Ron?
Sorry, I thought I'd done that. Hello, Ian. I seem to recall that there was another species of Margaritifera, which was Auricularia, which occurred as a fossil in this country, and as far as I'm aware, is still extant in Southern Europe. I wondered if you'd got any knowledge about whether there's any conservation work going on for this species, and whether that's been extensively fished for pearls in the past.
I'll answer your last bit first. I don't know, I'm not aware of it being fished for pearls. But yes, there are certainly still extant populations in northern Spain, in Aragon, and the Charente, Vienne catchments in central western France. None of the populations are huge, but there's certainly a lot of work going on both in France and Spain to conserve this species, catchment management, and I believe also to do some work on captive breeding. Sadly, one of the pioneers of that work, Rafa Araujo, died a couple of weeks ago. So that was very sad because he was a real pioneer of Margaritifera conservation as a whole, but particularly for highlighting all the issues with Margaritifera auricularia.
Okay, thank you.
Martin, last question, and then I think we'll move on. Can you unmute Martin, please?
Yep, I was under the impression that in Spain and I thought it was Portugal, certainly Spain, that some Durabensis auricularia populations were threatened by corbicular fluminea infestations.
I don't doubt it.
Yeah, I think they definitely are.
Very quickly, Matt.
Yes, one question. Do you have how many other species of freshwater mussels except corbicula do you have in the US, we have a lot of different freshwater mussels, and we're having to try to keep all those different species living.
I think we reckon there's about 15 species in Europe. And I know there's current work going on, which means we might have a few more in the not too distant future, once the genetics of these populations has been worked out.
How about in Scotland and London, England, and the UK?
In England, we have 4 lowland species.
Okay.
5.
Thank you very much, Ian. I'm quite sure there could have been more questions, but please can we all thank Ian for his splendid talk.